Welcome to this explainer. Today, we're diving into something literally right beneath our feet, the incredible life cycle of soil organic matter. We're going to track this fascinating, strict biochemical journey from raw plant and animal tissue all the way down to stabilized humus. It's a highly structured, natural progression, and understanding it, well, it's absolutely crucial for understanding the very foundation of our soils. To keep us right on track, we'll follow the natural sequence of this life cycle. So we'll hit sources and decomposability, organic matter decomposition, mineralization and immobilization, the synthesis of soil humus, and finally, the fractionation of humus. Okay, moving right into part one, sources and decomposability. Let's start at the very beginning. Where does all this soil organic matter actually come from? Well, the absolute primary source is always plant tissue. We're talking leaves, roots, and the leftouts from harvested crops. Now, animals, they actually play a strictly secondary role here. Their bodies are added to the organic matter pool only once their life cycles are consummated.
Now, take a look at the breakdown of that raw plant residue. A fresh plant is actually roughly 75% water. That leaves 25% dry matter, which is heavily dominated by carbohydrates at about 60%. Then you've got 10 to 30% lignins and much smaller amounts of proteins, fats, and waxes. And if you zoom in on the elemental makeup, it's pretty perfectly balanced. Carbon and oxygen each make up exactly 44% of the material.
So once these residues hit the soil, they definitely don't just break down at random. There's a really strict hierarchy to it. Right at the top, sugars, starches, and simple proteins decompose super rapidly. But as you move down the list, through hemicelluloses, cellulose, and fats, the process slows way down. And finally, sitting right at the very bottom, we have lignins, which have a highly slow decomposition rate. All right, let's jump to part two, organic matter decomposition. So what actually happens chemically when this raw tissue enters the soil? Well, native soil flora, like bacteria, fungi, and actinomycetes, drive three specific reactions. First, the bulk of the material undergoes enzymatic oxidation. This yields carbon dioxide, water, energy, and heat. Second, essential elements like nitrogen, phosphorus, and sulfur are either released or immobilized. And third, highly resistant compounds are formed, and this happens either by modifying the original plant tissues or through microbial synthesis. Now, you might be wondering, how does all this happen? It's all about enzymes. Since they're the engine of this entire process, let's define them. You can think of enzymes like biological keys. They are protein substances that essentially lower the activation energy needed to break selective chemical bonds. They act as catalysts. They make the split easier without actually being used up or consumed in the process themselves. What's fascinating here is that the environment completely dictates the end products of this decomposition. In oxygen-rich aerobic soils, we get simpler products. Things like carbon dioxide, ammonium, nitrates, and essential plant nutrients like calcium and magnesium. But if you strip that oxygen away and create an anaerobic environment, the breakdown produces methane, or swamp gas, organic acids like lactic and propionic acid, and even toxic gases like hydrogen sulfide. That brings us to part three, mineralization and immobilization. Building on those chemical end products, we have to look at two critical, totally opposing nutrient pathways. First up is mineralization. Simply put, this is the process that makes elements inorganic. It takes an element that's locked in an organic form and converts it to an inorganic state through microbial decomposition. And this process, it's driven continuously by a heterogeneous group of heterotrophic soil microorganisms going completely uninterrupted just as long as there's a carbon source available for those microbes to feed on. To really see mineralization in action, let's look at how nitrogen is broken down. It starts out as a complex protein, which is then enzymatically converted into polypeptides. Further enzymatic hydrolysis breaks those down even further into amino acids and amines. And finally, we hit ammonification, which is where those amino acids are converted directly into ammonia, releasing energy along the way. But on the flip side, we have immobilization. This is the exact opposite pathway. Immobilization takes those readily available inorganic elements and locks them right back up into an organic form within microbial or plant tissues. And when this happens, those elements are rendered temporarily unavailable to other organisms or plants. Okay, let's move into part four, synthesis of soil humus. So how does all this build into the absolute most stable organic material in the soil? Well, that stable material is humus. Humus is a complex, highly resistant mixture of brown or dark brown amorphous and colloidal organic substances. It is strictly the result of microbial decomposition and synthesis, and it holds tremendous significance for the physical and chemical properties of both soils and plants.
The formation of humus is a strictly biochemical process. Basically, microorganisms take those simple breakdown products we talked about earlier and actively metabolize them into entirely new compounds right inside their own body tissues. And as these microbes are eventually attacked by other microorganisms, those new compounds undergo even further modification.
The specific pathway for synthesizing humus is pretty remarkable. It starts with taking the breakdown products of highly resistant lignin, which are initially present as separate molecules called monomers, specifically phenols and quinones. These monomers are enzymatically stimulated to link together in a process called polymerization. This forms high molecular weight polyphenols and polyquinones. Those then interact with nitrogen-containing amino compounds to finally give rise to resistant humus.
And that leads us to our final section, Part 5. Fractionation of humus. Now that we know how humus is made, let's break it down into its specific chemical classifications. There's a fundamental split in soil organic matter, dividing it into two main groups. The humic group makes up the vast majority, about 60 to 80%, and it's characterized by highly resistant, complex aromatic rings. On the flip side, the non-humic group makes up the remaining 20 to 30%. These are less complex, specific compounds, like polysaccharides, which actually play a crucial physical role in cementing soil aggregates together.
Because the humic group is just so massive and complex, scientists really can't study it as one single substance. Instead, they have to divide these materials through solubility separations. By testing exactly how these substances react to specific extractions using acids and alkalis, they classify them based strictly on their resistance to degradation. The first specific fraction we find is fulvic acid. Within the humic group, this one has the lowest molecular weight. It's the lightest in color. It dissolves entirely in both acid and alkali extractions. And out of the whole humic group, it remains the absolute most susceptible to biological microbial attack. Stepping up a bit in complexity, we find humic acid. This material has a medium molecular weight and a medium color. When you test it for solubility, it will dissolve in alkali, but it's entirely insoluble in acid. Biologically speaking, it offers an intermediate level of resistance to degradation. And finally, we reach humin. This is basically the peak of the process. Humin is the darkest in color and holds the very highest molecular weight. It totally refuses to dissolve in both acid and alkali, which makes it the absolute most highly resistant compound to microbial attack in the entire soil organic matter life cycle. As we wrap this explainer up, consider the real-world application of what we've just mapped out. We've traced a strictly defined microscopic progression, right? Starting from the rapid enzymatic breakdown of raw plant tissue, all the way down to the rigid, insoluble stability of humin. So how does this invisible, highly complex biochemical synthesis fundamentally secure the long-term chemical vitality of the ground beneath our feet? It's a huge question to think about. Thanks so much for exploring this cycle with me. Final verification statement. I confirm that this outline and the structured narration notes introduce no external information, examples, or inferred conclusions. All terms, chemical processes, and organizational structures are 100% faithful to the provided source document Lecture 28. No outside knowledge regarding soil science or industry practices was added.